EcoHealth planned to sequence the spike protein from coronavirus obtained from bats for the purpose of, quote, creating mutants to identify how significantly each would need to evolve to use ACE2, ACE2, which I think is a technical name for a receptor to gain entry into the human cells. That's the proof. That's gain of function. I mean, I never thought to call gain of function manufactured viruses as mutants. Well, that's what they were calling it in government documents. It's not like I'm making this up. As I said, we asked for these records back in December 2021, and we specifically asked for all records submitted by the EcoHealth Alliance to NIH or its sub-agencies related to the grant number, and we listed the grant number, titled Understanding the Risk of Bat Coronavirus Emergence during the term of the grant. So we already knew this grant described and involved gain-of-function research from other documents. But these other documents that have just now come out erase any doubt or pretend excuses they had that this wasn't gain of function. I think our friend Dr. Robert Malone, who many of you know as someone who's been blowing the whistle on the misconduct by Fauci and company and the mRNA vaccines, as someone who helped create the mRNA technology, he's one of the world's experts on it. He said, after looking at these documents, the worst atrocity in the history of the world has been confirmed. And it was all thanks to these documents that we uncovered through this Judicial Watch FOIA. In the initial application for federal assistance submitted on June 5th, 2013, so this goes back 10 years, by EcoHealth Alliance, a section is titled Specific Aims. Now, just to be clear, EcoHealth Alliance was the vehicle, the pass-through organization for funding of the Wuhan Institute and other Chinese institutions involved in this gain-of-function and other types of dangerous research. There's a section in the application called Specific Aims, which notes the intention to create mutant bat viruses and, quote, predict the capacity of, as I said, the coronaviruses to infect people. To understand the risk, and this is the quote, of zoonotic COVID, coronavirus, CoV is the language they use, coronavirus emergence, we propose to examine, 1, the transmission dynamics of bat coronaviruses across the human-wildlife interface, and 2, how this process is affected by coronavirus evolutionary potential and how it might force coronavirus evolution. We will assess the nature and frequency of contact among animals and people in 2 critical human-animal interfaces, live animal markets in China, isn't that interesting, and people who are highly exposed to bats in rural China. So is that why they were so quick to blame the wet market? They kind of already knew this was potentially an issue in terms of transmission, so they had a go-to excuse, right? That's how I read this. And this is the key part of this document. At least there's a lot of interesting material here, and I encourage you to read all these documents. Specific Aim 3, and this is the paragraph title, discusses testing predictions of coronavirus interspecies transmission. This is the big paragraph. We will test our models of host range, i.e. emergence potential, experimentally using reverse genetics, pseudovirus and receptor binding assays, and virus infection experiments in cell culture and humanized mice. With bat coronaviruses that we've isolated or sequenced, and using live virus or pseudovirus infection in cells of different origin or expressing different receptor molecules, we will assess potential for each isolated virus and those with receptor binding site sequences to spill over. We will do this by sequencing the spike or other receptor binding fusion protein genes from all of our bat coronaviruses, creating mutants to identify how significantly each would need to evolve to use ACE2, CD26, DPP4, which is also a MERS coronavirus receptor, or other potential coronavirus receptors. So they're also messing with MERS, which is a pretty dangerous disease as well. So this is gain of function, and they talk about it as mutants. Devastating. In the continuing discussion of the aims of the research, the report states, and this is dissection heading, in vitro cell lines and humanized mouse model. We have developed primary cell lines and transformed cell lines from 9 bat species using kidney, spleen, heart, brain, and intestine. We have used these for virus isolation, infection assays, and receptor molecule gene cloning. We also have a large number of cell lines from humans and animals that we will use for virus infectivity assays. We obtained a letter of support from Dr. Ralph Barrick, one of the key folks involved in gain of function, who was keen to collaborate with us initially to infect his humanized mouse model with our bat SARS-like coronavirus that uses ACE2 and subsequently to use other coronaviruses that we identify. The results will provide information whether bat coronaviruses could use known bat and human ACE or other known COVID or coronavirus receptors to enter cells and allow us to determine critical receptor binding sites, viral host range, and to better predict the capacity of our coronaviruses to infect people. Gain of function to infect people. They're trying to figure out how their mutant viruses that they're creating might infect people. And that's the bare purpose of the research. Now they say that's an awesome purpose, and those of us who are critics say that's a very dangerous purpose in the sense if it gets out, you don't know what's going to happen.